Hello, welcome to EdHead's Genetics Lab. I understand you are here to help with some genetic counseling and DNA work we are doing here. Glad you could join us. I'm a bit swamped today, so I'm just going to assign you one of my cases today and you can jump right in. I'm a genetic counselor. I help people understand or adapt to genetic disorders or counsel people that might be at risk for inherited diseases. And that's what you will be doing today. We have a couple that is concerned that they have a genetic disease running in their family. They are considering children in the near future and want to know more so they can make an informed decision about their future. I need for you to review their case history and then report back to me so we can decide what to do. The Williams are an African American couple, and they each report a family history of sickle cell anemia. So what is sickle cell anemia? It's a disease where the hemoglobin protein molecules in the red blood cells are not formed correctly. Hemoglobin normally carries oxygen from the lungs to all the cells of the body. In sickle cell anemia, hemoglobin forms long fibers. Hemoglobin in these fibers does not carry oxygen as well as normal hemoglobin. The fibers cause the red blood cells to be sickle-shaped instead of round, so they don't go through blood vessels as well, or sometimes not at all. This can cause blockages in the blood vessels. This disease can be very serious and even fatal, so parents are right to be concerned about their children if this runs in their family. Hemoglobin in sickle cell patients is not normal because their DNA is different from individuals without the disease. This difference is called a mutation. Now that you have some level of familiarity with our patients, what would be the most logical next step? A. Do a DNA test on the husband. B. Do a DNA test on the wife. C. Do a family history of both parents. D. Let them know what treatment is available for the disease. Right. Our first step is to determine what the risk is of the couple having a child with the disease. Their risk can be determined by who in their family has the disease. This disease is recessive, so the adults could be carriers with one allele or gene that codes for the disease and one that doesn't. So let's have them do a family history and then take a look at what's there. So I've gotten the family histories of the couple. Let's take a look and see what we see. You let me know if you think they might have a child affected by the diseases we are discussing. Click on the genogram you would like to look at first. When you are finished with both, click Continue. Yes, that is correct. The family history does indicate that this person could be a carrier of the disease. If they have children with another carrier, the children could have the disease. Yes, that is correct. The family history does indicate that this person could be a carrier of the disease. If they have children with another carrier, the children could have the disease. Looks like these two people were smart to seek genetic counseling. They both have a history of this genetic disease in their families. So, our next step will be to run some tests to see if they are indeed carriers of the recessive gene for sickle cell anemia. But before we start testing, let's review what it is we are looking for here. The disease this couple is concerned about results from a mutation of DNA that creates abnormal hemoglobin protein. DNA is, of course, deoxyribonucleic acid. It is the genetic code that is passed from parents to children. So let's take a closer look at this code. It is normally found in a double helix that can look pretty complicated. Let's straighten that out to examine it more closely. DNA is composed of a nucleotide, which is composed of a nitrogenous base, adenine, guanine, cytosine, or thymine, a 5-carbon sugar deoxyribose in DNA, and one phosphate group. As you can see, thymine, T, fits perfectly and is always found paired with adenine, A. In the same way, cytosine, C, is always found paired with guanine, G. If you know half of the DNA, then you can build the other half because A is always paired with T. T is always paired with A, C with G, and G with C. You simply have to match the bases. Great! You know how to build a DNA molecule. But that is only the beginning of having a functional cell or organism. Just like you can build many cars from one blueprint, the cell can make many copies of a protein like hemoglobin from one region of DNA. Proteins are chains of smaller molecules called amino acids. There are only 20 different amino acids, yet when the 20 amino acids are put together in different sequences and different lengths, they make a spectacular variety of proteins in our body. The sequence of DNA bases is the code for the sequence of amino acids in a protein. 3 rungs of the DNA ladder is the code for one amino acid, and this code indicates the order the amino acids are linked together in a protein. It's a pretty simple code, but a very powerful result. Here is a chart of the triplet codes and the amino acids they correspond to. Note that there are several different triplet codes that correspond to one amino acid. Before a protein can be created, a process called transcription and translation needs to take place, involving messenger RNA and transfer RNA. This is a pretty fascinating topic all by itself, but isn't really what we're talking about here. So if you aren't up to speed on those processes, you might want to research them yourself. So as we said before, one triplet of DNA bases equals one amino acid in the protein. The amino acids of the protein have to be put together in the correct order for the protein to function. If the coding on the DNA is wrong, then the protein ends up being wrong too, and that's how you end up with a disease like sickle cell anemia. Amino acids are the building blocks of proteins, and all have a central or alpha carbon to which are bonded 4 groups: a hydrogen, an amino group, a carboxyl group, a unique side chain also known as a R group. The diagram you are looking at is the amino acid known as alanine. In sickle cell, there is one mutation or change in the DNA on chromosome 11. So assemble a segment of the normal protein, and then assemble a segment of the amino acids in the sickle cell protein. Great! You know how to build a DNA molecule. But that is only the beginning of having a functional cell or organism. Just like you can build many cars from one blueprint, the cell can make many copies of a protein like hemoglobin from one region of DNA. Proteins are chains of smaller molecules called amino acids. There are only 20 different amino acids, yet when the 20 amino acids are put together in different sequences and different lengths, they make a spectacular variety of proteins in our body. The sequence of DNA bases is the code for the sequence of amino acids in a protein. 3 rungs of the DNA ladder is the code for one amino acid, and this code indicates the order the amino acids are linked together in a protein. It's a pretty simple code, but a very powerful result. Here is a chart of the triplet codes and the amino acids they correspond to. Note that there are several different triplet codes that correspond to one amino acid. Before a protein can be created, a process called transcription and translation needs to take place, involving messenger RNA and transfer RNA. This is a pretty fascinating topic all by itself, but isn't really what we're talking about here. So if you aren't up to speed on those processes, you might want to research them yourself. So as we said before, one triplet of DNA bases equals one amino acid in the protein. The amino acids of the protein have to be put together in the correct order for the protein to function. If the coding on the DNA is wrong, then the protein ends up being wrong too, and that's how you end up with a disease like sickle cell anemia. Amino acids are the building blocks of proteins, and all have a central or alpha carbon to which are bonded 4 groups: a hydrogen, an amino group, a carboxyl group, a unique side chain also known as a R group. The diagram you are looking at is the amino acid known as alanine. In sickle cell, there is one mutation or change in the DNA on chromosome 11. So assemble a segment of the normal protein, and then assemble a segment of the amino acids in the sickle cell protein. So you can see clearly how the mutation changes the protein. The single substitution of valine for glutamic acid means that the hemoglobin protein behaves very differently. Let's take a closer look at how that might happen. Hemoglobin is a protein found in red blood cells that binds to oxygen and transports it around the body. There are about 280 million hemoglobin molecules in each red blood cell. Hemoglobin is composed of 4 chains of amino acids. 2 are called alpha chains, and 2 are known as beta amino acid chains. In most sickle cell patients, the disease is caused when one and only one amino acid out of the 146 amino acids in the beta chain differs from normal individuals. As you have seen, glutamic acid is replaced by valine. In hemoglobin, as with all proteins, different parts of the amino acid chains bind to other parts, holding the protein in just the right shape to do its job. This process is called protein folding. The bonds between amino acids and their R groups also control interactions between protein molecules, and this is what goes wrong in sickle cell patients. Compared to most chemical interactions, the forces that hold a protein in its folded state, or control protein to protein interactions, are weak. However, many weak interactions combine to eventually lock the protein in the folded structure. Because folding of most proteins involves many different interactions, The process is complicated, and even one amino acid change can drastically alter the overall result. There are only a few types of interactions leading to folding: hydrogen bonds, charge-charge interactions, or salt bridges, hydrophobic interactions, and disulfide bridges. Here is a protein with all of its atoms shown and a graphic of the amino acid chain. The primary structure of the protein is the sequence of amino acids in a chain. The secondary structure of the protein is made up of commonly found structural features, like alpha helices, beta sheets, and hairpin loops. The tertiary structure is the arrangement of the secondary structures, like the beta sheets and hairpin loops, into a final arrangement. So let's take a closer look at that list of amino acid interactions. A hydrogen bond is formed when a hydrogen atom links two atoms that are usually either oxygen, red, or nitrogen, blue. Here are two hydrogen bonds, shown as dashed blue lines, holding the beta sheet together. Some amino acids contain oily groups, like the benzene ring of phenylalanine and the double ring of tryptophan. Just like oil separates from water, these groups tend to fold to the interior of a protein, away from the water. These are hydrophobic interactions. A tryptophan and phenylalanine buried in the center of the folded protein are shown here. A salt bridge, also known as a charge-to-charge interaction, is easy to understand. The positive charge of one amino acid is attracted to the negative charge of another. Cysteine is an amino acid containing sulfur atoms. Two cysteines can form a strong covalent bond joining their sulfur atoms, which is known as a disulfide bridge. So given what you now know about protein folding, can you determine which bonds would form to cause the protein to fold? Can you name the type of interaction? Now you understand some of the interactions that drive proteins to fold, a very complex process. We've simplified the process by only showing a few types of amino acids. Try folding a protein with 55 amino acids. The blue amino acids can form hydrogen bonds. They will be close to each other, and on the outside of the folded protein where they can also hydrogen bond with water. This particular protein begins folding by forming an alpha helix, which is a secondary structural unit. One of the hallmarks of an alpha helix is bonding between amino acids that are in units of 4 along the chain. Look for a region with a repeating pattern of 4 amino acids. Click on these units to form the alpha helix. Great! Now you have the alpha helix. Note how the amino acids in the helix interact with others that are in the groups of 4. The green amino acids are the oily or hydrophobic ones. They will prefer to associate with each other and be on the inside of the protein away from water. The next step in the way this protein folds is called hydrophobic collapse. Identify 2 hydrophobic regions in the molecule. Good job! Notice how the hydrophobic amino acids form an oil drop in what will be the center of the protein. Also, note that the hydrophobic amino acids of the alpha helix face the center of the protein away from the water surrounding it. The final step is formation of a salt bridge, or a charge-to-charge interaction. Find two electrically charged amino acids. Excellent! Now you can see how proteins fold based on the amino acids that make up the chains. In the final structure, hydrogen bonding and charged amino acids tend to be on the outside, and hydrophobic amino acids are on the inside. The same principles apply to interactions between two different protein molecules. As we will see, hydrophobic interactions lead to the harmful behavior of hemoglobin in sickle cell disease. In sickle cell disease, an oily valine replaces a charged glutamic acid in hemoglobin. This small change in one of the beta chains, or one of the 4 chains in the molecule, introduces unwanted hydrophobic attractions between different hemoglobin molecules. Because the sickle cell hemoglobin protein folds differently than it should, it creates sticky places that cause the hemoglobin to create chains or fibers. It's these fibers that cause the red blood cell to be sickle-shaped instead of round, as it should be. That is how a single point mutation can create a lack of functional red blood cells in sickle cell anemia. The sickle-shaped cells die much more quickly than normal red blood cells. The sickle-shaped cells can also congregate in the small blood vessels of the body, particularly the hands and feet, causing them to be very painful and swollen. The oddly shaped cells also damage the spleen and increase the likelihood of infection and can significantly delay growth and development in children. Our job is to determine how best to counsel this couple regarding any children they may have in the future. We have agreed that they have family histories that indicate they might be carriers. So now we need to test to determine if they are both carriers and therefore at increased risk to have a child with the disease. Once we get the results of our tests, we'll do some Punnett squares to determine the likelihood of a child of theirs having the disease, being a carrier of the disease, or not being a carrier at all. So let's start with Robert Williams. Take a look at normal sequence for this gene on chromosome 11 and compare that to the sequence Mr. Williams has. Now let's look at Mrs. Jennifer Williams' DNA to determine if she is a carrier or not. What are your results? Right. Now we know this couple could potentially have a child with sickle cell anemia. Let's determine what the odds are for each child to have the disease, be a carrier, or inherit 2 functional copies of the gene. Drag the appropriate genotype into each box. Fill in the likelihood of each scenario for each child this couple might have. Okay, now we know what the likelihood is for a child of this couple to have the disease, to carry it, or to inherit 2 working copies of the gene. There is one more thing to consider. Carriers of sickle cell could show some minor signs of the disease, but not to the degree that a child with the disease, inheriting 2 non-functional copies of the gene, would. So, what do we tell the couple? Don't have any kids? There's a 75% chance something could be wrong. Have kids? There's a 75% chance nothing will be wrong. If you decide to have children, there is a 1 in 4, 25% chance the child could have sickle cell, and a 2 in 4, 50% chance that the child could be a carrier, and a 1 in 4, 25% chance the child would not even be a carrier of the disease. The decision is yours. This answer gives them the facts, but remember that there are other ways we might help this couple. Don't just give them numbers and walk away leaving them to struggle on their own. That is not what genetic counselors do. Good answer. We should sit down and discuss with the Williams exactly what sickle cell anemia is, how it is treated, and what the odds are to have a child with the disease. While it is a serious, lifelong medical condition, there are additional options for treating the child and making sure he or she has the best medical attention possible after birth if needed. People with sickle cell that get good medical treatment throughout their lives are living into their 60s and beyond. There are options for testing before a pregnancy, during a pregnancy, or after a child is born. There is a possibility for having a pre-implantation genetic diagnosis that would ensure the baby does not have a genetic disease. There are also options we can offer them for testing during pregnancy, like chronic villi testing or amniocentesis. The main reasons for testing during pregnancy would be to determine whether or not to continue the pregnancy or to have definitive answers to help prepare for after the child is born. A child could also have a blood test shortly after birth. Thanks for your help today. I appreciate you helping this couple by giving them the right information to make a very important decision in their lives. Also, there are tons of unanswered questions in genetics and genetic counseling that I hope you will consider researching in your future career. Thanks again.